Hello, today we're going to discuss some errors in meiosis. So, before we get into those errors, we first need to know what a karyotype is. So a karyotype is the number and visual appearance of chromosomes in the cell nuclei of an organism or species. So essentially, a karyotype is just laying out all the chromosomes of the species just to be observed. So in the image above, that is a human karyotype. So there's 46 chromosomes in humans, 23 pairs of chromosomes, the last pair being the sex chromosomes. And males have an XY chromosome pairing, while females have an XX chromosome. All the other chromosomes, so chromosome pairings 1 through 22, those are not sex chromosomes, and they are known as autosomes, which is another name for non-sex chromosomes. Autosomes. One of the main errors that can happen in meiosis is called nondisjunction. So it is the failure of one or more pairs of homologous chromosomes or sister chromatid to separate normally during nuclear division, usually resulting in an abnormal distribution of chromosomes in the daughter nuclei. So looking at this image now, the normal meiotic division with metaphase has those chromosomes perfectly lined up in the middle, and then they are split evenly across straight down. With nondisjunction, basically that even split is uneven. More chromosomes are pulled towards one cell or not enough pulled towards the new cell or new daughter nuclei to be formed. Now there's two different kinds of nondisjunction. There's nondisjunction 1 and nondisjunction 2. Nondisjunction 1 happens in meiosis 1, nondisjunction 2 happens in meiosis 2. And basically, that just indicates when those chromosomes are unevenly divided or separated from one another. When you look at nondisjunction meiosis 1, the end result is 4 cells with the wrong chromosome number, 2 cells with too many chromosomes, and 2 cells with not enough chromosomes in them. But if nondisjunction happens in meiosis 2, you get 1 cell that's got too many chromosomes, 1 cell that's not enough, and 2 cells that would still have the normal amount of genetic information. So both cases of nondisjunction can cause some serious problems, but nondisjunction meiosis 1 is worse than meiosis 2 because it creates more cells with the wrong chromosome. One example of what actually can happen when you have nondisjunction is basically, say you have a sperm or egg cell with the wrong amount of chromosomes in it, so either an extra chromosome or not enough. That pairs up with a sperm or egg cell with the appropriate amount, and you end up getting a fertilized, now zygote that has either an extra chromosome or is lacking a chromosome. And those cause some serious issues. Most of the time, when you have an extra chromosome or a missing chromosome, when the sperm and egg come together, it usually doesn't even result in a pregnancy just because there's too much incompatibility. But one of the more common examples of where pregnancy does still happen is when you have an extra chromosome on chromosome pairing 21. So now instead of a pairing, it's a triplicate or 3 chromosomes, and you can see that in the image on the right. And that is Down syndrome. There's a ton of problems with Down syndrome, one being intellectual disability and some other physical health problems as well. Now with Turner syndrome, you have similar effects as Down syndrome, except the effects are more centered around the individual sex, because if you have Turner syndrome, you only have one sex chromosome being the X chromosome. So you don't have another X or another Y chromosome. So there's a lot of identity factors that are incredibly detrimental to mental health because, physical identity might not match up with the mental identity or literally just the physical development as well. And you just have other health problems thrown in there as well. And also I want to point out that before I showed what an XY pairing would be for a male, so there you can see an XX pairing of chromosomes for female. Okay, the last thing is besides non-disjunction, you can have several different kinds of chromosomal mutations. So these affect much more genetic information than the chromatin mutations that were previously discussed. So again, chromatin is just uncoiled DNA, chromosomes are coiled DNA. So when you have chromosomal mutations, it's affecting whole chunks of chromosomes, which means it is just affecting so much more code of DNA, which is going to affect so much more proteins made, which is going to cause just a slew of problems. So looking from going from left to right, an inversion chromosomal mutation is where literally a part of the chromosome is flipped backwards. So all proteins would be read essentially in backwards code from what they're supposed to be. So you have a lot of wrong proteins produced there. Deletion, you just have a whole bunch of genetic information just mutated to be gone. So a lot of proteins that are needed to be made simply won't be made. Insertion is the opposite, so you can have one part of a chromosome be inserted onto a completely different chromosome. And basically, it's not that those proteins will never be read, but it's that they'll never be read at the time they are needed to be or when they are needed to be because they're located on a completely different chromosome now, completely different part of the DNA that there's just a bunch of communication problems there as far as protein signaling as to when to make proteins. And then translocation, you have the same problem, except it's basically two parts of two different chromosomes switch places with one another. As you can see, that kind of pink chromosome ends up on the yellow chromosome and vice versa. And that causes a lot of protein changes there as well. Thank you for watching. Please share and subscribe. And remember to spread